Good afternoon, everybody, and welcome to another one of our TESS seminars. And as you all know, TESS is our Center for Tropical, Environmental, and Sustainability Science here at JCU, which is one of our main bodies for promoting terrestrial and aquatic research, not marine research, but terrestrial and aquatic work. And we have around 200 people that are affiliated with TESS across the campus and outside the campus. So we're pretty happy about that. It's a real pleasure today to introduce Nigel Tucker. Nigel's been one of these speakers we've been wanting to have here for a while, and it's fantastic that Nigel's been able to make time. He's got a lot of things going on in his life, and he's very busy, but he's made time to come talk to us now. Nigel is one of the real recognized experts on restoration ecology and forest restoration and that type of thing. He's worked very extensively in North Queensland. He's done a lot of work with Steve Goosum, who's here with us today. And they've published at least 2 books that I know of on restoration and science and ecology in the wet tropics region. Nigel's also worked a great deal in New Guinea and in also the Southeast Asian region. For 20 years, he was the director of Queensland National Park's tropical herbarium up on the Atherton tableland, and they led a very active restoration, forest restoration and replanting program up there. Nigel was extremely involved in that and in promoting a lot of that work. And then subsequent to that, Nigel was always one of these people who was so dynamic doing so many things. You sort of wondered, like, you know, would any bureaucracy, would any agency be able to contain him? And he left that eventually and opened up his own consultancy company, which was very successful, Biotropica. And they did a lot of work in New Guinea and other places, Australia and Southeast Asia as well. So Nigel is a person with an enormous amount of field experience, enormous amount of practical experience. He's well published, very well studied, and we're just delighted to have him here today to talk about his 3 corridor project, which he's leading, exploring the outcomes of restoring habitat connectivity on the Atherton Uplands. Nigel Tucker. Oh. How's that?
How's that? The hat came in useful for something other than protecting my head, Annabelle. Welcome, everybody. Thanks very much for taking the time to come along this afternoon. It would be really remiss of me not to, at the very start of this talk, point out the fact that the inspiration for this work pretty much rests with Bill and Sue. You know, Bill and Sue have obviously been around in North Queensland for a long time. And in another life, Sue and I worked for the department from the National Parks and Wildlife Service, and Bill was with the Wet Tropics Management Authority. And I guess the work that Bill was doing on fragmentation and Sue's work on population biology really stimulated me to think about how the 2 sciences of conservation biology, which is a science of scarcity, and restoration ecology, the science of replenishment, could really work together much more effectively. So what I'm going to talk about today is the 3 Corridors project, which is a project involving myself, Dr. David Ting, and Dr. Amanda Freeman, and a range of others. And it would be remiss of me not to, at this point, also point out the fact that these people are equally responsible for pulling off this really great project. So we know that fragmentation and connectivity acting synergistically. We know that they're responsible for diversity declining at a range of scales. There's plenty of evidence of that. And some of the work that obviously Steve Williams has done with birds in the last 10 years or so really shows that up. It's interesting to me that improving landscape connectivity is the most frequently cited recommendation for climate change adaptation, which makes sense as well. But in terms of fragmentation, it's clear that we have a lot of fragments that are sitting out in a sea of farmland, and the long-term survival of those fragments really depends on ensuring that they're part of a well-connected landscape. So on the Atherton tablelands, I'm sure you'd all be aware that the tablelands is one of these really fragmented parts of the Australian landscape. Lakes Eacham and Barrine are both around 500 hectares. The Curtain Fig's around 300 hectares. They've all been isolated fragments essentially since the 1940s. They were just part of that movement of preserving little blocks of forest for their scenic attributes. And of course, what's left now is for us to try and manage them in a way that preserves the biodiversity that's contained within each. So between 1995 and 2014, community groups, volunteers undertook to restore connectivity between Lake Eacham, Lake Barrine and the Curtain Fig, joining them to Gadgarra, which is part of the Wooroonurin State Forest. So Wooroonurin National Park. So let's just have a look at the kind of things that I'm going to be talking about. This is a slide from a tree planting in 1995. This is at Donaghy's Corridor, and this is the site in 2024. A site in 1998, same place, and the vegetation in 2024. There are three corridors that I'm going to talk about today. So let's have a look at the way these are laid out. So at the top right-hand side of the screen is Gadgarra National Park. As I said, Gadgarra is part of Wooroonurin, which is an 80,000 hectare block encompassing Bartlefrere, Belland and Kerr. It's the largest block on this side of the river. 1 kilometre distant from Gadgarra is Lake Barrine, another one of these, or one of the three fragments. And joining them together is Donaghy's Corridor. Up here, Lakes Corridor joins Lake Barrine to Lake Eacham. You can see that there. And over on the far left, that's Curtain Fig National Park, 300 hectare fragment of Marby Forest. And it's joined now to Lake Eacham along Peterson Creek. Just quickly, some of the differences between these three corridors. First of all, you'll see that Donaghy's is actually unbroken. Lakes Corridor has a minor road traversing. Peterson Creek Corridor has a major road and a minor road. There are varying levels of disconnection, if you like, in each of the corridors. One of the problems is Bill's told me many times is, the landscapes of the real world make these kind of studies a bitch because nothing's a perfect replicate. As Bill said, it would be great if there were 20 corridors linking 20 fragments of the same size. It would be the perfect kind of experiment, but that's not the way the real world works. One thing I did want to just point out to you is we've got a range of ages. So Donaghy's Corridor was all planted in the 90s, between 95 and 98. Lakes Corridor between, in fact, let's just go to the next slide. As I said, Lake Barrine to Gadgarra, the Donaghy's Corridors, Sure.
Ellen, could you please turn your camera off?
1 kilometre long. It was planted between 96 and 98, involved around 17,000 plants. The Lakes Corridor joins Lake Barrine to Lake Eacham, 1.4 kilometres, around 8 years to establish and 17,000 plants. And Peterson Creek, the longest one at 4.4 kilometres, just over 100,000 plants to establish it. And you see the number of species which have been used in each as well. One of the reasons why there are more in Peterson Creek is obviously at Lake Eacham, we have regional ecosystem 7.8.2. When we get to Curtain Fig, we have regional ecosystem 7.8.3. So there is a transition of vegetation types between those two sites as well. So these Lake Barrine to Gadgarra, the Donaghy's and the Peterson Creek Corridor were all done by volunteers, members of TREAT working with the Queensland Parks and Wildlife Service. And the Lakes Corridor was done by the Lander Lake Eacham Land Care Group. working with the wet tropics tree planting scheme at Malanda.
We'll hold the questions till the end, please. Just use a microphone. So the question will be heard by the audience online.
Okay, so just a quick survey outline. We studied vegetation. We looked at the corridor vegetation. We looked at reference forest vegetation. We looked at regrowth that was in the corridors as well, to look at the structural connectivity which had been achieved over the decades. We did a bird and mammal survey over a 12-month period to look at bird and mammal communities in reference forest and in the corridors. And of course, to look at functional resilience, which is, one of these difficult things to study. It's like, what do you study? How do you study it? When do you measure it? Et cetera, et cetera. But if we're going to think about how functional these restored areas are, what kind of functional resilience do they have? We used a soil seed bank study as a surrogate to look at what might happen in the face of a massive disturbance to this vegetation. The vegetation survey, we looked at 57 transects, 50 metres by 3 metres, 36 in the corridors. And you can see the breakup there. There were 14 in old growth or old restoration, 9 in mid-age restoration, 13 in young restoration, 8 in regrowth, and 13 in reference forests. In those plots, we identified and measured all stems greater than 1 centimetre DBH, and we also did their height. We had 5 1-metre subplots where we studied the basal diameter of all stems. We counted seedlings. We did cover estimates of soil, litter, grass, dicotyledonous herbs, and canopy foliage projective cover. We also looked at coarse woody debris. And then at the end, all species were identified within the 50 by 3. And obviously, there's a lot of difference between a 10-year-old restoration plot and a mature forest. You can appreciate the differences, I'm sure. So this is an ordination of site floristics based on basal area. And you're going to actually see some really similar ordinations throughout this presentation. Look at the way the reference forest sites cluster together, the regrowth clusters together unbelievably closely. A nice cluster of old and then young as well. That clustering of the regrowth is quite exceptional, but it's a fact. But the interesting thing to me was that most of the old sites clustered together, most of the young sites clustered together, but look at the difference, the huge gulf between the two. So this is what Donaghy's corridor looks like after 25 years. This is one of David Ting's profile diagrams. You can see all of this vegetation here was planted, and all of this vegetation here has what's come up since it was planted in the 90s. To summarise the vegetation, what we found was the corridor vegetation structurally similar to reference forest, but differs significantly in terms of species composition, as you would expect, and diversity. What we find is that there are many, many more gap phase species in the corridors. Mature phase species are less common, and of course the opposite is true in the reference forest, where there are very, very low numbers of gap phase species and very, very high numbers of mature phase species. What's interesting is how birds and mammals have actually reacted to this vegetation. We, I guess, and I probably am just as guilty as the rest of us, of having a fairly anthropocentric view of this, is look, what would it feel like if I was an animal, would I like it? And what it turns out is that my perception is a little bit different to the way the natural world does see it. Let's have a look at birds first of all. This is Amanda Freeman's work, and Amanda, if you're listening, and I'm sure you are, Amanda is joining us, so that if anyone has any questions on birds, Amanda will be happy to answer. She actually had cataracts removed just the other day, so she's not in a state to drive and couldn't be here today. But Amanda established 22.3 hectares plots, and she conducted 6 by 30-minute seen and heard surveys in a seasonal survey over a 12-month period. Amanda classified her birds into functional groups and by their preferred habitat type. So the outcomes were over 2,000 records of 70 species, around 40 to 45 in each of the corridors and in the reference forest as well. But habitat type, planting age, and proximity to reference forest all had an effect on birds. This is a great diagram that shows changes over time. And I think changes over time and proximity, again, will be two of the things that you will see fairly commonly today. First of all, Peterson Creek, and this is from a range of different surveys. You can see that the number of endemics, regional wet tropics endemics, the dark green, it's been on the marginal increase, certainly at Peterson Creek and lakes. The delay in surveys between 2008 and 2022 really showed up at Donaghey's, but you can see that there are, over time, gradual increases both of rainforest-dependent species and endemics as well. The effect of proximity on these climate-sensitive species from Williams and De La Fuente 2021. This is a really interesting piece of work that shows how proximity is really affecting those climate-sensitive species. That they are certainly hesitant to move, and there are large numbers to be found within a kilometre or so, but the numbers really steeply decline from that point. And this also shows pretty much the same thing. You see the clustering in these reference forest areas, and also, you know, like Donaghey's 1995, 1996, and 1998, they're only planted 12 months apart. But there's obviously some really significant differences just between 98 and 95. And the 1995 plot close to Gadgara, obviously there's a lot of similarity in the communities that are there, including things like chowchillas. Within the corridor, and obviously chowchillas being one of those species which is really typically associated with mature forest. So to summarise the birds, proximity and age are obviously the main factors that are influencing the bird community structure in corridors. Abundance and diversity are both higher, closer to reference forest, especially for those climate-sensitive species. And endemic species are more common in old restoration sites. And there's a couple of whipbirds in that image that happen to be taken during the mammal surveys. But I think one thing I would just leave you with is this point here that endemic species are more common in old restoration sites. So let's move on to look at mammals. We set up baited cameras 100 metres apart in reference forests, in the corridors and in reference forests as well. We set them for 4 nights and 5 days. We did 4 seasonal surveys in wet, dry, cold and hot for 1,152 trap nights. All of the images were identified to species, and we also totaled the number of daily detections of each species as well. And for those of you who heard Matt Luskin's talk quite recently, those detections are a really important way of quantifying mammal visitation. And finally, we classified all the mammals as per the Wet Tropics Vertebrate Atlas, where 0 is for non-rainforest species and 6 is for species who rely completely on rainforest as habitat. So we're talking things like feral pig at 0 and a musky rat kangaroo at 6. The survey outcomes. We recorded 17 mammals, of which 4 were exotic, and an echidna. Yeah, the echidna is, in fact, I have to say, just as an aside, all of the cameras that I've had out in all of the corridors that I've been studying for a long time now, echidnas turn up every single time. And in winter, they're very common, as you can imagine. But I had never really, well, you know, I hadn't really thought about monotremes that much in fact, but it was interesting that it was actually present in every single survey at virtually every single site. What we found though was that VAC6 species are very uncommon, and in fact, the only VAC6 species that we recorded were the musky rat kangaroo and Lumholtz tree kangaroo. VAC 4 and 5 were actually dominant in corridors and in reference forests. What we found was that grassland rodents are now absent, and I guess for the first few years, I should just have mentioned that when we were establishing these corridors, in some corridors, we did actually sample for mammals, birds, reptiles prior to establishing vegetation. In 2 of these corridors, Peterson Creek and Donaghy's corridor, for the first 2 to 3 years, we had very large numbers of of Mellamy's burtonii, Burton's climbing rat. And at around year 2, Burton's climbing rat was joined by its cogenitor, Mellamy's savinipes, the fawn-footed millipes. And by year 3, Burton's climbing rat, the grassland species, had disappeared, and it had been taken over by Mellamy's savinipes, the forest species. So it was no real surprise that in all of the studies in all of the images that we took, there were no grassland species at all. And I'm talking about things like Rattus sordidus, Mellamy's burtonii, Mus musculus, those species that we would normally associate with grassland habitats. Finally, white-tailed rats, bush rats, Cape York rats, which I'm sorry, I should mention, we didn't distinguish between Cape York rats and bush rats, and you wouldn't try using a camera, obviously. But those 4 species, the white-tailed rat, bush rat, Cape York rat, and fawn-footed Mellamys, they have virtually continuous populations. That is to say, they were sampled at every single 100-metre point in all 3 corridors. I wrestled with whether or not I should just pull this slide out, but I'm going to leave it in for a couple of reasons. First of all, to show you that VAC6 species are uncommon. In fact, the tree kangaroo was only detected in Peterson Creek corridor. We know, obviously, it's over at Curtain Fig, but from looking at iNaturalist records over the last few years, I note that there were very, very few, in fact, 0 records of tree kangaroos at Lake Eacham until 2013. And since 2013, there's been a significant number of tree kangaroo sightings at Lake Barrine. And in the 20 years that at Lake Eacham. In the 20 years that I worked at Lake Eacham, I certainly didn't ever see a tree kangaroo. And there's a really interesting image on iNaturalist of a tree kangaroo crossing the road at Lake Eacham from the Curtain Fig area. So none of that, it's all obviously anecdotal, but the fact that the tree kangaroos detected in the corridor at about halfway along Peterson Creek corridor, so it's about 2.5 kilometres, so it's virtually in the middle of the corridor. The other one, the musky rat kangaroo, it was picked up in Donaghy's corridor, Lake's corridor, and in the 3 7.8.2 reference forest sites. I'm not, I wouldn't expect to see it at Curtain Fig. I'm pretty sure it wouldn't occur there naturally. So it's no great surprise to see it not present at Peterson Creek, and a really refreshing site at both Donaghy's and Lakes. The rest of these 4 and 5s, you can see they're pretty well distributed through corridors and reference forests. The one thing I would draw your attention to is this single record of Dendrolagus, this 2 records of the coppery brushtail, and 3 records of the paddy melon. And all of those species have the heaviest body weight. They are all the largest bodied species that you would expect to see in, apart from, of all the mammals, they are the heaviest body weight, and obviously in very low numbers. All of the 3s, you can see here in the corridors, but in fairly low numbers, but most, they're just almost completely absent from reference forest sites. Just before I show you the next slide, these singletons of the rabbit, they're very low numbers of the pig. They tended to skew the ordination that Bill helped me with, so I've left them out. You have a look at this ordination, you'll see that, again, most of the reference forest sites are clustered in pretty well here. This is obviously where things like the Hypsiprimnodon and Thylogale are appearing as well. In the centre, you can see these really common Rattus fuscipes, Melames, Savinipes, and Urames. And over on the left-hand side, Dendrolagus, the tree kangaroo, and of course, the coppery brushtail. No great surprises there. In terms of age and proximity, I'd like you to just spend a little bit of time looking at this one. First of all, what I've done is broken each corridor up into constituent parts. That is, the bits that are close to reference forests, the bits that are more distant, and the bits that are in the middle. At Peterson Creek, you can see that the 2 ends of the corridor have both got young vegetation, and the number of species and the number of detections is quite low. In the middle of the corridor, which is actually where the oldest parts of the restoration plantings are, is where tree kangaroos and the most detections were recorded. And I say this because what we found with the birds was that there were more endemic species in the oldest plantings. And no surprise that what we saw at Peterson Creek, what we see at Lakes, and what we see at Donaghy's is the most diverse and the most rainforest-like communities are all in the oldest parts of the planting. So what's happened here at Peterson is that animals have been prepared to traverse through suboptimal vegetation, through younger parts of each corridor to get to the better quality older habitats that are more distant from reference forest. And I really want to just emphasize how important these ground mammals like musky rat kangaroos and white-tailed rats are. This is a schematic of Donaghy's corridor. When we're doing veg surveys there, we came across a number of species that are large fruited. Yellow walnut, black walnut, and cassowary plum. These are all fruits that weigh between 125 and 250 grams. They're all sort of small cricket ball, large golf ball size. And they all, as I said, weigh up to 250 grams. So here is the nearest known fruiting cassowary plum. 400 meters away is a cassowary plum seedling. Down here, we've got Endiandra insignis, gully laurel. And we've picked up 1, 2, 3, 4 seedlings of that gully laurel, the most distant being about 130 meters away from the parent tree. And obviously, when we encountered these things, we did really complete 100-meter surveys around all of these seedlings to make sure that that was the closest possible parent. Now, the parent may be further away, but that's as close as it possibly can be. So most interestingly, we had in here and in here seedlings of Bosmedia bancroftii, the yellow walnut. Again, one of those things between 125 and 250 grams. But there are no plantings or pre-existing stems of Bosmedia bancroftii. There are no stems of Bosmedia bancroftii within 100 meters of those seedlings. So they have been brought in, I suspect, by a white-tailed rat, because obviously a musky rat kangaroo is not going to traverse this pasture, because this is all totally surrounded by pasture. So somehow or other, 6 individuals of these Bosmedia here and 3 up there have been brought into the corridor by either a musky red kangaroo or a white-tailed rat. That has important implications, because for those of us who've always expected that rats do nothing but predate seeds, that's obviously bullshit. But the other thing is that it is not only cassowaries that are potentially going to be moving these things around, is that there are other animals which are quite capable of not only persisting in these fragments, but moving seeds from reference forests and other fragments on the landscape into these corridor settings. So to wrap up on the ground mammals, obviously age and proximity again are major influences. Rainforest obligates, that is the VAC 6 species, they occur in the old restoration sites, regrowth and reference forests, but they're virtually absent from young and mid-age sites. Individual responses by these VAC5s, things like musky red kangaroo, like paddy melons and white-tailed rats, are much more varied. And as I've just explained, VA6 are really functionally important in the dispersal of large fruited species, like the Cryptocarya oblata. The final aspect of the study that I wanted to talk about is a soil seed bank study. As I mentioned, we're interested in looking at the corridors from the perspective of how functionally resilient are they. So if we think about things like floods, cyclones, droughts, pretty normal kind of events in the forest ecology in North Queensland, what effect do they have on restored areas? We know that obviously Cyclone Larry and Cyclone Yazi have shown us that forests do bounce back, but what we were most concerned about was what's the ratio of weeds to the ratio of native plants in the soil seed bank. That might give us some kind of reference point in terms of how recovery might occur. So we conducted a seedling emergence study. That is, we collected soil and we germinated it. We took 131 cores in pasture, corridor, and reference forest. So we were interested in looking at what's going on in the pasture. Are birds dispersing seeds into the corridor margins as well? We retained the soil in the cores completely, and they were watered twice a day at the QPWS nursery at Lake Eacham. We did a species ID and seedling removal at 30, 60, and 90 days, and seedlings were classified as either native or exotic and by life form. Outcomes. We got 1,427 seedlings from 27 families, 32 were native, 37 were exotic. The small seeded pioneers like Acacia, Alphatonia, Ficus, Congesta, etc., they're very common in both corridor and reference forest samples. Pastures were completely dominated by grasses and forbs. Figs, as in ficus, was the dominant native genus. 7 species, 78 individuals, including, climax species like Ficus watkinsiana, Ficus pluricarpa that you would only expect to see in mature phase forests. We found significant differences between the site types, that is pasture, rainforest, corridor. But most pleasingly, the exotic species that we recorded in the corridor samples turned out to be mostly benign. We look at life forms. You see in the middle there, the forest is really, the reference forest samples were really dominated by native trees, especially gap species, a couple of canopy species, but very, very few exotics. To the right of the forest is obviously a dominant forb, grass, herb community in the pastures. And over on the left, the corridor sites, which are kind of an amalgamation of both. And obviously they all differed as per X squared. I think, yeah, look, I'll come to that. Age and proximity had even an effect on soil seed banks. You can see that in terms of the year of planting, this is just some generalized linear modeling, the volume decreases significantly as we move both away from the forest and in terms of age. The older the planting, the more diverse the soil seed bank, and the more distant from forest, the more depauperate the soil seed bank. And an NMDS pretty much confirms all of that, showing that the 3 groups are quite dissimilar. And to summarise, the corridor soil seed banks are more similar to reference forest and pasture. Exotic species dominate pasture seed banks. Exotic species in reference forests and corridors are mostly benign. They've got very little ability to outcompete the woody native species that are there. And finally, gap species and ficus are common soil seed bank components, indicating their attractiveness to frugivorous dispersers. One thing I would also mention, which was quite an unexpected outcome of this, which I haven't shown you here. Most soil seed bank studies, there is a correlation between, there is no correlation between above ground and below ground vegetation. So the trees that you see, they are quite similar to the seeds which are in the soil seed bank. That held true for the reference forest samples, because in the reference forest, we had a canopy of mature rainforest and a soil seed bank, which was comprised almost entirely of pioneers, that is acacias and alphitonias, etc. In the corridors, we had lots and lots of pioneer seeds in the soil seed bank as well. What was interesting was those species formed the canopy. And again, in the pasture, all of the seeds that were in the soil seed bank were exactly the same as the plants that were growing above it. So there was a strong correlation between standing vegetation and the soil seed bank in the corridors and in pasture, but quite a different correlation, no correlation at all, in fact, in the reference forest. So to finalise, that image of the antechinus on the bait, it's like, it's mine. It's mine and it's all mine and I'm not going anywhere. But let's summarise the talk. First of all, vegetation, reference forests and corridors is structurally similar, but differs significantly in terms of composition and diversity. Proximity and planting age are structuring influences on birds and mammal communities and the soil seed bank. the presence of endemic species in older, more distant sites confirms two things. It confirms movement and it confirms colonisation. And I say movement because, as I pointed out, animals have obviously traversed pretty suboptimal habitats in order to get to the best habitat, which is the oldest habitat and is not always the habitat which is closest to reference forest. And finally, seed banks indicate that these corridors have achieved a semblance of biological memory that will allow them to recover in much the same way that reference forest would recover post disturbances like droughts, floods, or cyclones. So that concludes my talk, and I would love to answer any questions that anybody might have.
I'm a very interested, very much outsider, so this is the first time I've heard any of all this stuff. So please excuse me if I've got a dumb question. But you mentioned the two mammals that might have moved those seeds away from the trees. Are there no cassowaries in that area? Why were you excluding cassowaries?
Good point. And I mean, I'm not excluding cassowaries. I would actually, if you'd asked me that question 10 years ago, I would have said, oh no, look, there's just no chance. But in the last few months, there's been reports of a couple of cassowaries at Lake Eacham and a cassowary at Lake Barrine as well. Now, I don't know how they got there. Maybe they came through the corridor, maybe they came through a more hospitable matrix than the matrix that was there 30 or 40 years ago. But my point is this. I wouldn't expect cassowaries to eat Bosmedia bancroftii or Endiandra insignis. They're not cassowary food plants. The cassowary plum, I agree 100%. And the fact that it's gone 400 metres across pasture, from pasture into the corridor, is just one of those enigmas that I really don't know. I mean, it could be a flying fox. But, you know, cassowary plum, that's a heavy piece of fruit. You know, to be a pretty fit, strong flying fox that would contemplate carrying something like that 400 metres. But in answer to your question, yeah, those other two are not cassowary food plants, but they are certainly food plants of musky rat kangaroos and white-tailed rats.
Any other questions? Yep.
I was interested in your comment re the musky kangaroo and the white-tailed rat moving the seeds. Anecdotally, talking to all the other people up there, are they sort of indicating the same thing?
Look, there's been a couple of published studies. A guy by the name of Thyma published some really interesting work looking at musky rat, looking at white-tailed rats and their movement of those two species, Endiandra insignis and Bosmedia bancroftii. Thyma found that both of them were being moved by white-tailed rats, and he didn't record them moving the kind of distances that we have, judging by the seedling germination. But I guess this is, you know, it's a very good question because it kind of throws some sort of traditionally held beliefs a little bit to the side. I mean, for instance, if we go back to this one here, these, you know, this is pasture. These things are being, the corridor and those fragments up on the top of the screen, they're separated by 130 odd metres of grass that's about that tall. So, I mean, I would like to think that a musky rat kangaroo would, but he just wouldn't. I mean, it just would not be in the realms of possibility. But for a white-tailed rat to move something that heavy over that distance, yeah, that I'd, you know, I mean, that's not stretching my level of incredulity, but it's coming close. I mean, that's, sorry?
You might have eaten half of it first.
Well, no, that's a good point. But this is the other thing, is that they discard the fleshy covering. They're only interested in the tester, the hard bit of the seed. So even if they did, they still had to, strip all the stuff off and have a snack halfway down. And if they did that, then obviously it's not going to germinate when they get it the rest of the way. So they've got to take it whole. They've got to cache it, bury it, without destroying it, without putting any kind of incision into it. So, as I said, if I hadn't seen it myself, I would be still questioning anybody who said that they'd seen or had an example of a white-tailed rat carrying something up to 250 grams, 130 meters through grass. The only other way it could have got there was from gravity. But it's not going to roll through grass.
Oh, okay. Sorry, yeah, go ahead online.
Oh, good day,
Elliot?
Nigel. Elliot here.
Hey, Elliot.
Hey, quick question. We know that roads create barriers and divide these habitats up and can change your animal movements, and not to mention wildlife vehicle collisions. Have your studies recorded a difference in the diversity of fauna either side of the Malanda Lake Barrine Road? And do you think that the canopy rope bridge that's there makes a significant difference? Because we know that we're noting that some animals have been captured on the camera traps using that rope bridge to cross that road. But do you think that that rope bridge actually makes a significant difference to the diversity of fauna either side of that road?
Yeah, I think it definitely does. I mean, I suppose one of the issues, and yeah, you'd be particularly understanding of this, Elliot. One of the problems, as I said at the very start, is that connectivity is a very difficult thing to measure unless you're just talking about a wildlife overpass where you're going to put a camera at either end, because there are so many variables, because it's so difficult to get to avoid pseudo replication, to get a number of replicates that are that make it statistically viable and believable. So one of the things which is really well studied up here are the rope ladders, and the one we put in it to cross the road from Lake Eacham over the Lake Barrine Road to get animals into the Peterson Creek corridor. We actually found white-tailed rats using that within about a month of it going up. And that was actually the first time that I've seen white-tailed rats using a rope bridge. Some surveys were done in that area at around the same time, and they turned up pogonymies, mollipilosis, the pogonymies, prehensile tailed rat. Now, the occurrence of white-tailed rats, pogonymies, and Lumholtz tree kangaroo, all in that area around where the rope ladder was put up, would seem to suggest that the road's a barrier, but it's not something that animals won't overcome. Obviously, Elliot, we would love to see Transport and Main Roads commit to doing some really great work on those culverts underneath the road, and I'm sure TMR would really love to do that. Hint, hint. Because I think the presence of water, the presence of vegetation on both sides of the road, all that does is elevate the importance of the site in terms of where animals are going to be concentrated. And so it does, by my reckoning, mean that anything which TMR chooses to do that improves the quality of habitat around the road and around those crossing points, and the culverts in particular, would be a really big benefit. Does that answer your question, Elliot?
Yeah, thanks. And can I follow that up with another question? Currently, the department has undertaken a project to create a wildlife corridor between Katana wetlands and the highlands, being Granda National Park. What lessons learned could you shed light on for design and implementation of this particular corridor?
Look, I think, you know, the landscape isn't a blank slate. There are always remnant trees here, little patches there. There are streams. All of these little microhabitats influence the way you might get animals to coalesce around a particular area. And I think using what's already there, using what's already there has been shown to be a big advantage. I think if I hark back to Donaghy's corridor, you see these fragments, these were there before the corridor was built, or when at the time the corridor was built. It's no surprise that the diversity of mammals and birds is highest around that area, because obviously having that vegetation there beforehand, there was a structure there, there was a source of food there, and obviously the stream is there as well. So to kind of pick up on Elliot's point, using the landscape, recognizing that there are features of the landscape that are already there and that can be used to enhance and to speed the process of recovery, I think is really important.
Yeah, thanks very much.
Amanda.
Oh, yes. Just wanted to make a comment on Elliot's question, following up. It's not to do with the rope bridges, but it is to do with the Malanda Lake Barrine Road. And that is that until very recently, we had no records of grey-headed robins between the Malanda Lake Barrine Road and Peramon Road. So in that central part of the Peterson Creek corridor. But recently I went back through Peterson Creek corridor and I did get grey-headed robins in that section of the corridor. And now I don't think they're using the rope bridge, but I do think it's a factor that now that Freeman's Forest has grown up right down to the edge of the road, and no relation, by the way. But now that that revegetation is so much better established, I think the road no longer poses a barrier to that species, whereas it did previously. That's my thought on that. So.
And I just wanted to bring your attention to that section of the Peterson Creek corridor. That's actually the oldest piece, the bit in the middle. So it had virtually probably a kilometre of grassland either side. And what's most interesting is that's where, so we're picking up tree kangaroos all through that area. And most recently, as I said, a crossing of the road at Lake Barrine as well.
You mentioned that the one species that you sort of found everywhere was echidnas. Do you think that'd be expected in the pastures as well, or is it just you're getting them more of their food in your corridors as well?
Yeah, I think you're right, that last point.
Yeah.
I think, you know, the fact that there's a more varied source of food,
yeah,
cover, would stand to reason.
Another question? Yeah, go ahead.
What you mentioned earlier about the dissimilarity of the standing timber to the seed bank, I found that in the reference forest. I found that quite interesting, and I was wondering if you have an idea of why that's the case. Like, I'm a bit surprised why there wouldn't be any mature seeds in the ground, and they can't be all dispersed and go away. And also, some of them would have a longer lifetime. Like, I'm aware that some piney species seeds just live much longer than the mature phase ones, but anyway.
Yeah, no, you're right on both counts. First of all, so it was interesting to me as well that, you know, the soil seed bank and the vegetation in the corridors was virtually the same. And I guess what's most interesting is, well, the reason that they're there is because they were planted. So when the planting was done, pioneer species generally take up between, say, 15 and 20% of everything that's planted. So things like acacia, alphatonia, tremor, ficus congesta, those Poliscius elegans, those kind of early successional pioneers, they're about 20% of everything that's planted. They're now the canopy. Sure, there are some other things that are the canopy as well, but essentially they're the canopy and they're what's depositing seeds into the soil seed bank. Most interesting was the figs. So I said that we got 7 species of figs and 78 individuals. What was most interesting to me was that 70 of those 78 individuals came from Donaghy's corridor and the Peterson Creek corridor. In the Lakes corridor, figs were not planted. And the only figs that turned up in the soil seed bank there were early successional species that have been brought in by birds, Ficus fraseri and Ficus congesta. In the other 2 corridors, where figs were about 20% of what was planted, and there were probably 7 or 8 species planted in both corridors, their seeds are in the soil seed bank. And interestingly, those figs also turned up in reference forest, but they didn't turn up at Lakes corridor where the species hadn't been planted. So that's showing that, okay, what's eating things is also dispersing those seeds straight in, and they're being incorporated into the soil seed bank. But what you see is what you get. The soil seed bank in those corridors is entirely comprised of species that were planted. Whereas in the reference forest, obviously, there were not only were there none in sight, but they were also super abundant in the soil seed bank. So essentially, what you plant has a big effect on the soil seed bank. If you don't plant pioneers, if you don't plant figs, the soil seed bank ends up being quite depauperate, because in lakes, what we found was that the density and the diversity was much, much lower than it was at Donaghy's and Peterson Creek, where figs hadn't been planted, and in fact, where very few pioneers had been planted as well. So there is a direct relationship between what you plant and what will be in the soil seed bank. So it kind of, to me, reinforces the importance of planting pioneers, not only as an important structuring group, but also as an important group in terms of being incorporated into the soil seed bank and being able to contribute to recovery post-disturbance.
Very good, yeah. I'll come right back.
Does that answer your question?
Not quite. I was actually interested in the reference forest. There's pioneer seeds in the ground where there's mature phase species.
Yeah, because mature phase species, the seeds have no longevity.
That's fine.
Yeah, they have, you know, those people who grow rainforest plants will know that once seeds get past six or so weeks of age, the germination decline goes right down. Whereas, you know, small seeds easily incorporated into the soil seed bank, like figs and alphitonias and acacias, and much more, obviously, that hard seed coat gives them the ability to hang there for years.
Like once medium and crofty, maybe a bit of an exception.
You certainly wouldn't expect to see Bosch medium and crofty in the soil seed bank. And just as an aside, the largest seed we had come up in the soil seed bank was a candle nut in the reference forest samples at Lake Eacham. And another pioneer, essentially. I mean, candle nut is not what you'd expect to see in a really, you know, it's an early successional species. But interesting that it was, you know, it's that size. So it's not that it can't be incorporated into the soil seed bank.
Yeah, when it survives the rats.
Yeah, exactly. Well, you know, the Bosch media and the Endiandra said to me, wow, you know, like rats are stupid, but they're endearingly stupid because they're doing us a great favor in terms of dispersing those things which normally would be immobile because their dispersers are lost.
Hi, yeah, great talk. It's obvious how much work's gone into this, and it's great to see all the mammals and birds and all the different plants starting to come back. I guess I'm just wondering, are there further plans to do more surveys with other sorts of organisms? I should preface here, I am an entomologist, so I'm a bit biased towards invertebrates.
Yeah. And look, I would have loved, I would have loved to have an invertebrate ecologist work with us on this, because the numbers that you can get are just so interesting. One of the things that we did really early back when Donaghy's corridor was first established, one of the things that we did was put logs all over the place. Like we'd get logs that had been sourced from all over the place, because obviously big pieces of dead would take a long time to arrive naturally. And then a JCU PhD by the name of Simon Grove, who now works in Tasmania, a great coleopterist. He and I did some surveys of those dead logs 12 months after the corridor had been established, and found a really remarkable suite of things that colonised those things already. There's a bucket load of, well, there isn't, unfortunately. There's a few papers out there that talk about colonisation, you know, Mia Durhay's stuff, most recently with dung beetles. Some of Peter Grimbacher's work with beetles themselves. Really interesting. And if you're ever interested in conducting some future studies like honours or PhD or whatever, then yeah, I'd love to talk to you.
Absolutely. Yeah, sounds good. Sounds good. I'm swamped under with my own PhD at the moment, but maybe in future. Yeah, thanks.
Sorry.
Do we have time for one more? No, they've already had their questions. So Nigel, I've been thinking a lot about the browsers.
Browsers, yeah.
And the thing is, because when we did our study on natural recovery, Omelanthus is really rare, but it's a big part of restoration planning for good reason. It's a wonderful plant. And I'm really curious that it is rare in natural recovery. And because predominantly, supposedly, the padymelons eat them.
Browsers, yeah.
But then it makes me wonder about everything else that we see, that we're counting. What are we missing because of all the browsers that are out there? And that we're only getting like a snapshot of what plants are recruiting.
Yeah, that's a good point.
Because we're not considering all the things that are getting eaten. And Omelanthus seems to me like a classic thing.
And there are others too. I guess, in fact, I can remember doing a planting at on Marby Forest near Atherton, and actually having to put up a fence around a hectare planting because the padymelons were savaging a narrow range, but savaging everything. And I have no doubt that in nature, they would be doing the same thing to those same plants. Absolutely.
Any other questions online or here? Okay, one more.
A question in two parts about your seed bank. One is, how deep did you take the soil samples? And then in the pastures where the cattle have been there for many, many years and you've got hard, compacted, trampled ground, did you actually find many good quality seeds in the pasture ground?
Okay, first part of the question, we used cores that were 100 millimetres across, 50 millimetres deep. But we know that most of the seeds are in that first 50 millimetres anyway. And after each, so what we'd do would be identify those germinants, pull them out and get rid of them, or pop them up if we didn't know what they were, and lightly till the soil to encourage more germination. The second part of your question, you'll have to remind me.
In the pastures with the compacted ground.
Again, a reflection of that what you see is what you get, not always. The effect, okay, so I should mention that there were some pasture samples that were collected from one part of the pasture which hadn't, from one part of a pasture which hadn't been grazed for some years. Now, there was a couple of pioneers growing in there, but nothing more than what we saw in pasture that was actively grazed. So there was no real difference. Whether it was ungrazed for 3 years or still grazed, the effect was the same. There was very little soil, very little native plant seed in the soil seed bank in the pasture. That could be an effect of trampling, but as I said, there was samples that we collected from pastures where cattle hadn't been for 3 years. The numbers were still the same. Which kind of, is interesting because you would expect that, birds would be, going to occasional trees that are outside the corridor and that you would see that kind of, but you don't. The other thing it shows is that these areas, before they were planted, there was not much there. You would think that, after 20 or 30 or 40 years of grazing, that there might still be something hanging around in the soil. But those soil samples showed that the pastures are totally devoid of anything that's going to encourage a regrowth.
Oh, one more.
And that's a big thing too, because a lot of us are interested in being able to use natural regeneration as a restoration tool, as a mechanism to stimulate recovery. And there's plenty of studies that show that in the rest of the world, in other parts of the world's tropics, that's quite a viable thing to do. But here, for some reason, the pace of recovery, the influence of things like guinea grass, slows down that natural regeneration significantly.
Still on the subject of the corridors, and I know this is not part of your study, but you may know from talking to the people who did do the planting, how much research would they have done? How scientific was their choice of what to plant? Oh, was you? Sorry.
And yeah, look, as I said, we would plant typically 15% pioneers. We would plant around 20% ficus, figs, because figs are such an important, species in tropical forests in terms of being able to provide frugivores with fruit at some time of the year when there isn't fruits available elsewhere. And the other 65% or so were fairly carefully selected mature phase species. And most of them with a very strong bias towards frugivory, because most of the plants, around 80% of all the plants in tropical forests, as Steve will tell you, are fleshy fruited and are dispersed by birds and mammals. So my approach was always to use that to try and drive the recovery process along by establishing a bait crop of things like bleeding heart, which produces those really nutritious fruits that can be eaten by most frugivorous birds. Those things that really drive the system along. I'd like to maybe close just by showing you a photograph. So that tree there is a bleeding heart, Homolanthus novaguineensis, and that was taken at Donaghy's corridor. So that tree was planted in 1998, and that photograph was taken in 2022. So there were 1,300 of that species planted in the corridor. Now, in all the surveys that I've done, that's the only one that's left. But those 1,300 plants have been replaced by 50 species which have come from the forest at either end. So over the course of 30 odd years, the planting has been enriched by an additional 50 species which have come from bird dispersal from forests at either end. So I'm not saying it works, but there was some really, there's over the last few years, there's been a lot of interesting studies done looking at the relationship between frugivory and dispersal and attracting birds into particular areas using fruits with particular qualities. And there's some really interesting work that's just been done in Brazil over the last few years that really highlights how important or how much you can improve or increase bird visitation just by choosing the right species.
Sorry to hog the show. At Science Week at the tanks one year, JCU scientists were there talking about what are that thing, pechacucha, where you get up and you talk for 20 minutes or 20 seconds or whatever it is. And there was somebody that you might know, but I can't remember who he is. And he was doing a study, I think in Borneo, of where they wanted to put a road through the rainforest. Was that
Probably
you?
Bill Lawrence.
Oh, sorry. Well, at least I remembered what you talked about. And I was fascinated that if it was a certain width, the bird wouldn't cross the thing.
And that's Susan's work, the lady sitting right behind her.
Small world.
Well,
It was Amazon, not Borneo.
oh, I'm sure. Well, maybe there was another one. This is Borneo. Anyhow, it made an impression on me. So I'm thinking about that in relation to your corridors as well, and the birds dispersing the seeds and so on.
Yeah, and that's one of the reasons why things like roads are really important, because obviously they disrupt. As Susan will tell you, there's some understory birds that just will never cross roads. They just will not leave the forest. Those birds, you know, birds that, for instance, can open their beak a long way to swallow a big seed, they're really important. And being able to encourage bird movement to the greatest possible extent is a really important part of tropical forest restoration.
Well, Nigel, thanks very much. Really interesting talk, relevant to lots of people. A lot of people are doing and thinking. So thank you very much. Really appreciate it. And thanks for coming down and joining us.